B cell develop in the bone marrow. So they start from a stem cell. They undergo so-called gene rearrangement, which I explain later. And they develop into individual B cell. And this B cell then migrate to the spleen and lymph node. And what's important to note is each B cell carry its unique antibody. So there might be around 1 billion B cells. You will have 1 billion different antibody. It's specific for their own antigens. So they are sitting in the lymph node and spleen and waiting for the antigen to come in. So if we see an antigen, so let's say a SARS-CoV-2 spike protein, yang COVID-19 itu, then this number 2 can proliferate, duplicate the 2. And then they duplicate 34, then duplicate become 8. And with repeated stimulations, the antibody on the cell surface will get secreted. So then become secreted antibody over here. And then at the same time, they can also undergo so-called class switching, changing the constant regions from IgM to IgG or to IgE or IgA. Okay. So IgM can have antibody also over here, but when repeated stimulation will cause them to become a different isotype, that's IgG, IgE, or IgA. So I think I say it again over here. There are more than 1 billion B cells, each with a different antibody on the membrane. There are very few naive B cells that's specific for SARS-CoV-2, so it's undetected. And because obviously, initially, you are not infected, but when you are infected, they'll proliferate. Only specific B cells that are specific for the SARS-CoV-2 will proliferate, and then you can detect it as an IgM positive. And further, it will detect it as IgG positive. So what's the difference with IgM and IgG, and what's the functions of all the different B cells? Well, antibody have 3 main functions. One is called neutralization. Neutralization ini yang paling penting untuk virus. Antibody will then bind to the virus, and you can see over here when the antibody bind to the virus, let's assume these are your SARS-CoV-2, the red one, SARS-CoV-2 virus. When the antibody bind there, it will block the virus from binding to the S2 receptor. So it cannot get in. And then once that happens, this whole complex can be engulfed by macrophages, another cell type that I'll discuss later, and it will just make it disappear. Another function is called opsonizations, where the antibody will coat, let's say, a parasite or bacteria, and this coating will allow macrophages to engulf the whole thing and again destroy it. And the last function is complement lysis. So antibody can cause the complement system, which is a group of protein, will punch a hole in the parasite membrane and kill the parasite. So these 3 different functions are the function of the antibody. There are also other functions like NK cells, activations, but these are the 3 main functions. And neutralization is the main functions for the virus. So here's a reason why there are different antibody isotype. Just focus on IgM and IgG. Human, actually, there are 4 different IgG, IgG1, 2, 3, and 4. There's neutralization. IgG is actually better than IgM. Opsonizations, IgG also is better. You can also sensitize the NK cell, which I also discussed later. This is natural killer cell. And again, IgG is better than IgM. Okay, IgE actually is for allergy. I'm not going to discuss it here. And IgA is let's say in the eyes or in the intestine. I'm not going to discuss it either. So IgG and IgM are the 2 things that I'll discuss. Okay, so how do you detect this IgM versus IgG? Well, I know in Indonesia, a lot of people use this rapid test. And so what are these rapid tests? So rapid test is basically, there is a pad over here where you conjugate or you put this colloidal gold over here is this circle that's red, and combine this with blue or green S protein from coronavirus, that's a spike protein. And then as a control over here, you also have a gold that's combined with a rabbit antibody. Okay, so this is a gold IgG rabbit antibody conjugate, and these are the gold that's conjugated to the S spike protein from some coronavirus. And then you have the M line, the G line, and the control line. So the M line to contain the anti-IgM antibody. So over here, this is antibody over here. Let's see, anti-human IgM antibody, in that's link over here. And in the G line, there is an anti-human IgG antibody. Okay, and then in the last line is the anti-rabbit antibody. So when you put your serum over here in the sample pad, it will flow up or flow whatever up or to the right over here. And if there is no antibody against the coronavirus protein, it will just flow and carry this rapid IgG with the gold, go all the way, carry all the way, and then it will stop over here. And it will stop over here because the anti-rapid IgG will capture it. The anti-IgG, rapid IgG antibody will capture this because it will bind to the rapid antibody, and the gold will cause it to be visualized as color over here. So you can visualize the gold. But if the serum obviously contains IgM for the virus, then in addition to carry this control, it will also carry this green one, which is the one that has the spike protein. You can see over here, the antibody from the human will bind to this S protein. They're carrying the gold. It will then stop over here because there is an anti-IgM antibody that will bind to the whole thing. So then it will cause color over here. The color will change in this stripe. And same thing with IgG. The IgG will go all the way to here and give you the color over here. Okay, so I actually said over here, colloidal gold can give color. It's conjugated to SARS-CoV-2 as protein, or the rapid IgG is placed on the conjugation pad. The M line contains the IgM antibody, the G line contains the IgG antibody, and the control line contains the rapid IgG antibody. Antibody in the blood that bind to the SARS-CoV-2 protein will carry the colloidal gold and stop the M for the IgM and stop in the G for the IgG.
Zoom.
Okay. So I know a lot of people in Indonesia use this a lot. And you can have different results, obviously. You can see here, this negative. If there are two lines here, means you're IgM positive. And if you have two lines over here, it's IgG positive. And you have three lines, is IgM and IgG positive. So obviously, very fast, cepat sekali, 15 minutes, cheap, so it's murah, and can be done without equipment. So bisa bikin tes ini di rumah. But a lot of con, it doesn't directly detect the virus. So remember, this only detects the antibody to the virus, so the indirect detections. And it misses the asymptomatic stage and early symptom stage. So these two stages actually is the stages where there are the most virus. So it basically misses the whole stages where the person is the most infectious. And it also gives you a lot of false positive. Why? Because remember, there's the other coronavirus that can cause flu ringan, common cold. So this antibody can cross-react, reaksi silang, and can bind to the S protein from coronavirus, from the SARS coronavirus, and give you false positive. So positive, but it's not true, the coronavirus. not COVID-19 coronavirus. It can also give you a false negative because they are not sensitive enough. The colloidal gold is not very sensitive, so you detect only with your eyes, and it can cause many different cases. You can miss many cases. And it's not quantitative. It's positive, negative, you don't know how much, and all these things. So a more accurate assay, I think the lab used a lot of this. It's called ELISA, or enzyme-linked immunoabsorbance assay. So basically, in these assays, you bind the antigen, in this case, spike protein, to the plate, to the well. So let's say this is the spike protein. And then you add the sera from the patients. Okay? And if this patient has the antibody against the spike protein, it will bind. Then you have to wash. So when you wash, you get rid of any non-specific binding. So this is a step that's not used in the rapid test. And this is an important step that can get rid of a lot of non-specific binding. And then you add good anti-human immunoglobulin antibody that has been conjugated to an enzyme, for example, horse peroxidase. Okay, so this is good anti-human antibody. And then, of course, you wash it again. And if there is an anti-spike antibody that's present in this well, the presence of this enzyme can be visualized if you add a substrate. In this case, for example, TMB substrate. This will emit light, in this case, yellow light. And you can then detect this by machines, spectrophotometer machines, and you can quantitate this. And basically, this is the basis for a lot of more accurate and more quantitative antibody tests that has been used, for example, by Roche. They use chemiluminescence. It's very similar, but instead of color, it gives you light. So the next two slides are the example of what happened if we use these ELISAs in COVID-19 patients. So each dot corresponding to a value from one human. And if you compare human that has symptom, yang ada gejala, dan human have no symptom or asymptomatic, or Indonesia namanya OTG, yang tidak ada gejala, IgM-nya hampir sama. So you have the same IgM level between these two groups of patients. But if you look at IgG, you can see that the one that has no symptom have a lot less IgG compared to the one that has symptom. Okay, maybe they have less virus, that's why they have no symptom. But then less virus means also less antibody. So the next one is against the use of this ELISA. In this case, it's a different scenario. This is a paper that was just published a few days ago. It's the vaccine study. So in this case, it's a vaccine from Oxford University, AstraZeneca company. They use adenovirus from chimpanzee that carry the spike proteins, and you inject them into human twice within 1 month. And then you measure the amount of the anti-spike antibody using ELISA. And this is human with no vaccine. So you can see here is the control. And again, each dot is the amount of the antibody in the human. And then if you give them the vaccine, you can start seeing this antibody in the beginning, there's almost nothing. You can see the antibody for the spike protein go up, and it can keep on going up as the days goes on. But you don't start seeing it until day 14, and then it would then peak around day 30, 35 and keep on going, and then maybe a little bit going down. And you can compare this with those that have the disease itself. So these are patients that have the SARS-CoV-2 virus, so COVID-19 patients. You can see the amount of antibody in this vaccination is somewhat similar to the antibody from the mild patients. Okay, so you can see over here too, green each dot is from a different human, green is from mild patients, and this one asymptomatic, you can see crosses, and the one that's severe also have more antibody. But basically, the vaccine is pretty good. It gives you antibody that's similar to those patients who have recovered. The one that has mild has already recovered, obviously, and similar amount of antibody as those that have recovered. So this is very promising that these vaccines may work because they have similar antibody amount as the one that has recovered. Okay, so why does it take so long? So that's the questions that I'm going to address quickly. Why does it take so long? Why does it take 2 weeks to get this antibody response? Well, there are two kinds of immunity, like Prof. Iris have said before. Innate immunity, or Immunitas bawaan, is fixed, is rapid, but very modest specificity. And innate immunity is mediated by infected cell yang udah kena infeksi, a group of cells called macrophages, dendritic cell, natural killer cell, and many other cell types that I'm not mentioning here. And then there is adaptive immunity, or Immunitas adaptive. Now, ini yang very slow, but it's very highly specific, and it has memory. It has memory means if you get infected again, it would seem to remember, oh, I got infection before. So then the response is faster and also stronger. And so this is the most important immunity. It's mediated by helper T cell that has a CD4 on the cell surface, B cell, B cell therapeutic B, that secrete antibody. So these are the ones that secrete antibody. And CD8 positive killer T cell, pembunuh cell T, that can kill viral infected cell. Okay, so ini gambarannya ini, B cell and T cell. So viral clearance, if you want to get rid of the virus, you really require the action of both innate and adaptive immunity. So innate immunity is very fast, but adaptive immunity is very slow. And that's why it takes some time, around 30 days, to get rid of this SARS-CoV-2 virus. So here's a quick comparison. Innate immunity takes 1 hour, a few hours to respond. Adaptive immunity takes days. You can see in the vaccine takes 14 days. And for IgM, it usually takes maybe 7 days. And IgG also 14 days. And the innate immunity is the same, whether you see it second time or third times. But adaptive immunity, it gets stronger and stronger. So if you get infected again, hopefully not, obviously, for the SARS-CoV-2. But if you get it again, maybe let's say in the vaccines, so the first vaccine, it would have antibody for the spike protein. So when the real virus come in, it would thought, oh, I've seen this before. So then the response will be stronger. So we call this a memory. So a receptor, there are receptor on all these innate immune cell that will see the pathogens, because then you can distinguish self from non-self. So for example, toll-like receptor, RIG-I, something like that. But none of these have any diversity. So they are the same in its cells. So toll-like receptor in, let's say, dendritic cell, one dendritic cell is the same, or to toll-like receptor in second dendritic cell, the same as the second dendritic cell, a third dendritic cell, etc. But for the B cell, it's different. Its B cell has their own antibody, very unique antibody. And same thing with T cell. T cell has a T receptor, and it's also unique for its T cell. So it's incredibly diverse. In fact, there are around 1 million different receptors for T cell, and for B cell, it could be 1 billion different receptor for T cell. So that's the most important aspect of the immune system, because that's how it can also basically get rid of a lot of other pathogens. You remember the pathogens in the environment, there are so many of them. And the ligand, so what the receptor see for the one for the immune cells, we call this pathogen-associated molecular patterns, like viral double-stranded RNA. But for B cell, which is the antibody, this antibody can see almost anything. And the one that you have to remember is the T cell receptor, or TCR here, only see MHC proteins in the context of the peptide. which is a part of the protein. So that's the difference. The main difference with T-cell receptor antibody. So antibody can see anything, T-cell receptor only see MHC and peptide. And that's the main reason why it takes so long to get adaptive immunity. Okay, so let's go back to the SARS-CoV-2. So here's a schematic diagram of what happened if you get SARS-CoV-2. So this is the lung. SARS-CoV-2 go through the lung. It will then bind to the H2 on the ciliated cell. And some of these receptor, rhytic I and toll-like receptor, will then see this, oh, there's some virus here. That's not good. So we'll try to get rid of this virus. And the way to get rid of this virus is to secrete this inflammatory cytokine. So, for example, this infected epithelial cell will secrete interferon. In this case, we just abbreviate IFN. And interferon can inhibit viral replications. So interferon will go back, it gets secreted, it will go back to the cell, will go inside and signal to inhibit the replication of the virus. So virus will not grow that much. Okay. And at the same time, interferon will also recruit macrophages and dendritic cell to this infected cell. So basically telling the body, I got a virus here, I need help, please come. So macrophages and dendritic cell will then come. But this virus is also very smart because it can also express the non-accessory protein. Remember, there are 16 different other proteins, and one of them actually can inhibit production of interferon. Okay, so then interferon production is actually pretty less because when you get infected with SARS-CoV-2, so then the effect of the interferon is a lot less than usual. So this infected cell will then also secrete other cytokines like TNF-alpha, IL-12, and other chemokine. IL-12 can activate this cell called NK cell, natural killer cell. This natural killer cell will also then kill this infected cell to get rid of the virus. So that's the only way to get rid of the virus is to kill all the infected cell. But that's not enough, okay? Macrophages will also secrete IL-1, IL-6, and TNF-alpha, and all of these work together trying to get rid of the virus that has infected this lung epithelial cell. And this all occur in the lung, in the airway wall. So innate immunity basically is not sufficient to clear the virus. In general, that's what happened. It's not just for the SARS-CoV-2, but for any virus. And the main reasons is infected epithelial cell cannot activate the adaptive immunity. So this basically is limited over here. It just cannot do it. It cannot activate the adaptive immunity that's required to get rid of the virus. So in the illustrative chart over here, you can see this is day after viral infections, the virus will replicate and keep on going and keep on going. And in this case, the interferon and cytokine will then be produced. It's transiently produced. This is the amount going up and then it come down. Natural killer cells start to go up and come down. So viral replication is limited. You know, if you don't have interferon or NK cell, the viral replications go down, just go up like this, like logarithmic, and just kill the human. So they do help, but you can see it's not clear. It's still some over here. And to get rid of this amount of virus, you really need to activate the adaptive immunity, which, starting around here, this you can see around day 5, and this adaptive immunity will then go up. And then as it goes up, it will then get rid of the virus. And you can see over here, the virus titer will become 0. So this is a very fast one. This is around 10 days, but for SARS-CoV-2, unfortunately, it takes around 30 days to get rid of the virus. And this adaptive immunity consists of antibody, but also helper T cells, and also specific killer T cells that can kill infected cells. Okay, so there are 3 arms of the adaptive immunity that's required to kill completely the viral infected cell. So how do you activate this adaptive immunity? Well, the key is a cell type called dendritic cells. Okay, so dendritic cells form the bridge between innate and adaptive immunity. So the key here is for the dendritic cell to acquire viral particle, maybe from cell that have been killed by NK cell. And dendritic cell will then activate CD4 positive, which is a helper T cell. And helper T cell will then help to activate B cell, as well as killer pembunuh T cell ini, pembunuh cell T, CD8 positive T cell. And then they'll go back to the lung. So all of this actually happen in the lymph node. So you can see how busy it is. The dendritic cell have to acquire the viral particle from the lung. It then have to migrate to the lymph node. And from the lymph node, it will activate the T cell. T cell activate the B cell, secrete antibody, and then this whole thing will have to go back, migrate back to the lung, and as well as the CD8 positive T cell then have to migrate to the lung and to get rid of all the viral infected cells. So it's a long process. So over here I showed you. Dendritic cells require to activate CD4 helper T cell, pembantu cell T. CD4 T cell is important for antibody and CD8 killer T cell productions. So without dendritic cell, there is no adaptive immunity. Without CD4 T cell, there is no adaptive immunity. So that's why it takes some time to get antibody. Basically, it's a very complex system before you can get the antibody productions. So why do you need dendritic cell? Well, dendritic cell are very special. Also macrophage and B cell. Immunologists call this antigen processing cell. They're special because they express two proteins. It's called P7, P7-2. Well, this is two, but we call it one protein. The other one is actually called MHC2 protein. Okay? So these two proteins are special because most cells, they don't have it. Like lung epithelial cells just don't have this protein at all. And because they don't have this protein, they cannot activate the T cell. Okay, so APC can activate the T cell. So here's a T cell. This is the APC like dendritic cells. They have the MHC2 that had a peptide, which I'll explain later, and P71, P72. So this MHC will bind to the T cell receptor on T cell, give you signal 1. This P71, P72 will bind to CD28 on T cell, give you signal 2. And sometimes they also will secrete cytokine that also help T cell to differentiate. So what's MHC? MHC stands for major histocompatibility complex. It has two classes. In humans, it's called HLA. It's very polymorphic. So different humans have different HLA type. It's the one that you need to match if you want to need transplantations. Of course, you need to match the blood type, but HLA is the one that's very, very difficult to match. And that's the one you need to match if you don't want an organ when you get organ transplant. If you don't want the organ to be rejected. So there are two different classes. MHC class 1 is expressed in all nucleated cells, so every cell except red blood cell. MHC2 are expressed only in this antigen processing cell, so dendritic cells, macrophages, and B cell. And they always have a peptide. So peptides are basically chunks of amino acids from the protein. So this peptide can be from self protein, but it can also be from non-self protein, so like the SARS-CoV-2 virus. Peptide will associate with both MHC1 and MHC2. And remember, T cell receptor only recognize this. T cell receptor from T cell, they don't recognize anything else. So when dendritic cells acquire the SARS-CoV-2 protein, this protein is then degraded into peptide. Okay, so this protein is chopped off into pieces. And this peptide is then loaded onto the MHC2, and the whole thing goes back to the membrane. And so now it can recruit T cell. So the T cell will then see this as something is wrong here. Or there's some peptide here that I never seen before. These are peptides that's not from self protein, but peptide from virus in this case. Okay? So interact with the T cell receptor, they are specific for them and deliver signal 1. So signal 1 from the MHC2 SARS-CoV-2 peptide on dendritic cells, will then activate T cell because also dendritic cell express B7-1, B7-2 that give you signal 2. So activate T cell, so in this case, it then can proliferate and secrete cytokine. So here's an example. Dendritic cells give you signal 1, signal 2, activate the T cell. Okay, T cell can grow. So this is a T cell that's specific for the SARS-CoV-2. It will then proliferate and then help to kill the viral infected cell. So the killer T cell express CD8 will then kill the viral infected cell because this viral infected cell will then express the peptide, the viral peptide with MHC1. And so you need the killer T cell for the you need the killer T cell to kill viral infected cell, and this killer T cell need to be activated by the helper T cell. And of course, B cells also need helper T cell. So then this helper T cell, one is activated by the dendritic cell, can give you a signal 2 in terms of cytokine, tell the B cell that recognize, say, a spike protein, okay, you can proliferate and you can secrete the antibody and help to kill the virus. So again, like T cell, B cell need 2 signal for activation. Signal 1 from the membrane-bound antibody that recognize the viral protein, and signal 2 from the CD4 positive T cell. Okay, so here's an example from severe patients. These are 10 severe COVID-19 patients. So on this first chart, this is the virus RNA detected by PCR. So you can see here, each line is actually, one line is actually one person. So this is patients during the next 30 days over here. You can see this patient is very lucky. The 10 days virus has already gone down to 0. Some patients are not as lucky. These patients still have virus at day 30. So these are all detected by PCR. And then you can detect the antibody, IgM or IgG in this case. They are specific for the virus. And you don't detect it until day 10 usually. Again, this patient is very lucky. You can see this actually at maybe day 4. You can all have IgG. Wow, this is a very strong human. So these are the amount of IgG that keep on going up. And as I told you, this IgG is important because then it will help to get this virus. But at the same time, you know, when you see IgG, you have to see the helper T cell. Because without the helper T cell, which has CD4 on the cell surface, you're not going to get an antibody. So of course you have IgG, but of course you also have CD4 positive T cell. Okay, so you can see the amount of virus-specific CD4 T cell going up and up also at the same time. So again, this is one human, this is the second human, there are 10 different patients. And here's the reference, by the way. If you want to read more about these papers, you can always refer to this reference. So all my slides has references that you want to have further study, you can read the papers. And the last one is the viral-specific CD8 positive T cell. It's actually, I find it interesting that not all the patients actually have it. So these patients, 2 patients have a lot of CD8 positive T cell, but many other patients actually don't have it for whatever reasons. And maybe that's why they have severe disease. And maybe some of these actually die eventually. And the one that have a lot of CD8 may actually recover. So that's the T cell response. And I know you're interested also in vaccines. So here is a graph from a vaccines produced by China, CanSino. This is also published just a few days ago. So in this vaccines, they use adenovirus 5. It's a common cold virus that contains the SARS-CoV-2 spike protein. And they inject it to the muscle. And then they measure, of course, they measure the antibody. But in this case, as I show you, the interferon gamma expressing specific T cell. So this is both CD4 and CD8. So you cannot distinguish between the 2 classes. So this is all the T cell. And they inject it at 3 different doses, low, middle, and high. So at day 0, you don't have any T cell that's specific for the virus. But by day 14, all the donors, or all the, not donors, or all the volunteers that have received these vaccines have T cell response. Okay, obviously the one that has high dose has a lot more T cell response. And then at day 28, it comes down a little bit. So that's normal, I guess. But they usually will maintain T cell for a long time. T cell can live for years, unlike B cells. Okay, so it's almost an hour already. But I only have maybe 10 more slides. So I just want to quickly summarize what I've told you so far. So lung epithelial cell express S2, which SARS-CoV-2 used to infect the cell. Infected lung epithelial cell secrete cytokine and chemokine to recruit many other cell types, dendritic cell, macrophages, and activate NK cell to kill themselves. Innate immunity is not sufficient to clear the virus, but you need to activate it, and you need these dendritic cells that are S2 negative. So dendritic cells have to somehow acquire the SARS-CoV-2 protein, which then activates CD4 positive helper T cell in the lymph node, and then activated helper T cell are required for activation of B cell and for CD8 positive killer T cell. And then antibody that's secreted can then neutralize the virus, and CD8 positive T cell can kill viral infected cell. And finally, you just clear the virus. Okay, so in the remaining of the talk, I just tell you about cytokine storm, and then I tell you about the more promising drug using antibody, which hopefully will be the magic drug to kill this virus. So as you can see on this chart, so in contrast to those patients which have mild symptoms, some patients become severe or critical. And these actually are also accompanied by so-called hyperinflammations. Okay, so instead of regular inflammations, these are very hyperinflammations. They have organ failures, and some of them actually die. And all these patients has elevated amount of inflammatory marker. So, for example, over here, you can see on the left is IL-6. Remember, this is a cytokine that I showed you earlier, and TNF alpha also. These are the amount of TNF alpha in mild patients, patient ringan, severe patients, and critical patients. And you can see the color over here. The mild patient, most of them have very low amount of TNF alpha. So this color actually shows you here a very less amount of TNF alpha. And when it's very, very dark, it has a lot of TNF alpha. So when the patient becomes severe, there are more TNF alpha amount in the serum. And in critical patient, you have even more, a large amount of TNF alpha. And same thing is true also with IL-6. So we call this hyperinflammations or cytokine storm. It's like a storm, cytokine, but coming in storm. It's lots of them. So this increased IL-6 and TNF alpha in critical and severe patient. It's a patient actually from Wuhan. Again, here are the papers if you are interested. So it's not completely clear why some patients develop cytokine storm and some don't. So one possibility which we are doing research of in my lab is maybe there are massive inflammatory cell death that's induced by the virus. So virus can induce cell death, and there are different kinds of cell death. Some are apoptotic, so they can get clear very easily, and some actually are so-called inflammatory cell death. The cell death causes inflammations. So the virus causes massive inflammations that can lead to hyperinflammations. On the other hand, virus may also get into the macrophages and then somehow stimulate virus to produce lots of cytokine. So remember, macrophages, they don't have S2. So as I told you, immune cells, they don't have S2. So virus cannot infect the immune cell directly, but it can maybe go in indirectly. So in this example, you want the antibody to neutralize the virus, but some antibody may not be neutralizing, and this antibody can actually help through a receptor called Fc receptor, will then go into macrophages and these macrophages will then infect, be so-called, then almost like getting infected, and these macrophages will then secrete a lot of the pro-inflammatory cytokine and become hyper-inflamed. So just give you a quick example. So cytokine, when it's present in normal level, like in this case, to get on my glasses here, IL-1, TNF-alpha, it can activate vascular endothelium, facilitate IgG and cell entry to tissues. Here's IL-6, it activates lymphocyte and antibody productions. It is IL-8 actually, also as a chemokine. It can recruit neutrophil, basophil, and T cell. IL-12 can activate NK cell, for example. So these are normal immune cells during immunity, a normal immune response. Right? But when you have systemic acute inflammatory cytokine storm, this large amount of IL-1, IL-6, and TNF-alpha can cause many disseminated effects. It can cause fluid loss, collapse of blood vessel, huge and rapid loss of blood pressures, clogging of blood vessel, shock, of course, which then leads to death. So in severe or critical COVID-19 patient, it's been shown that this can be partially mitigated by a drug called dexamethasone, which all of you know is an old drug. This drug is anti-inflammation. And maybe they can be mitigated by anti-IL-6 or Actemra, but this is still in clinical trials, so it's not really proven. So in the next slide here, I shows you what happened if you give this severe patient dexamethasone. Okay, these are charts. These are the paper. I don't remember where this come from, but anyway, patients who is very severe and they have to be under ventilator. And if you measure the mortality rate, unfortunately, you can see here, if you just give them usual care, some of them die day 7 after, basically this is randomization, so days after they decided to give some patient dexamethasone. So this is patient that have no dexamethasone. Some of them die, and if you follow them to 28 days, 40% of them unfortunately die. But if you give them dexamethasones, you can follow them, and only 30% of them die. So still die. So it's not a magic bullet, but a lot less than if you don't give them dexamethasone. So 40% versus 30%. But if the patient don't need oxygen, so that means they are severe but not really critical, give them the dexamethasone, actually did not do anything. Okay. Now, of course, you don't want to give them dexamethasone too early because you want the immune system to work. Right? So this is only when the immune system is out of control. Okay, so I just want to quickly tell you something that's more promising. So that's something that's looking forward instead of having no drug against this. Maybe there is an important drug, and so this is antibody for the SARS-CoV-2. So you can actually make the antibody, and if you can make in a large amount of this antibody, you can use this as a drug. And this has worked in against Ebola. So Ebola now is not as bad as before because you can give them antibody to Ebola to cure the patients. So hopefully this will also happen with those who have COVID-19. So this is a paper published by Regeneron. It's a company in the US. But there are 2 groups in China who also use the same thing, and there's another group in the US who also have come up with a lot of this antibody. But I just want to describe quickly how they make this antibody. So this Regeneron group have used a very interesting way to make this antibody, and they make it in humanized mice as well as convalescent humans. These are humans who have recovered dari orang yang sudah sembuh dari penyakit ini, and also in humanized mice. Okay, so antibody has been used as drug more recently, and it's very, very powerful and very, very useful. But the old way is very time consuming. So over here is an antibody from mouse, labeled in red. Here is antibody from human, labeled in blue. So you can make antibody specific for protein in mice, but if you inject this antibody into human, well, humans say, oh, there's something foreign here, and that's not good. So they'll then fight the antibody. So it will then elicit a lot of side effect, and that's not good. So people are very smart. So they say, okay, let's just replace the constant region with the human. So you have this so-called chimera antibody. So you replace the constant region, and of course the variable region has to be the same because that's the one you want. Okay. So for example, drug called Remicade, this is anti-CD20. This is a drug for non-B cell, non-Hodgkin lymphoma, B cell. Or Rituxan is anti-TNF alpha, drug for arthritis. These are chimeric antibody that's specific for CD20 here, and then the constant region are human. You can further replace the variable region with the human counterpart, and we call this humanized antibody. So example of this is Xolair, anti-IgE for allergy. Actemra, the anti-IL-6, also a humanized antibody. And then Avastin is actually anti-VEGF, is used for some cancers. Okay, so this antibody actually works pretty well, but it's still not completely human because there are some part that's mouse. And so there are two newer way and faster way to produce the human, completely human antibody. Okay, so you want antibody that's completely human. So the first way is from so-called humanized mice. And I explain to you what humanized mice are. So this an example, you can inject the SARS-CoV-2 spike recombinant protein to mice with the human antibody gene segment, so there's humanized mice, to obtain antibody with human variable region against spike. You then isolate the B cell using fluorescent activated cell sorter, which I explained. Perform single cell PCR to identify the variable gene segment for its B cell. Then produce the individual human antibody after fusing the variable regions to a human constant region, introduce them into cell line, then you can make lots of antibody. You can test the ability to block infections. And the second way is from peripheral blood lymphocyte or peripheral blood mononuclear cell from human that have recovered from this deadly disease. But you also use the same procedure as above. So basically, you take this PBMC, you isolate the B cell using fluorescent activated cell sorter, perform single PCR, then produce them by introduction into a cell line. So quickly, I could have to introduce some molecular biology here. So its antibody is produced from a rearranging genes. So the gene for antibody is not like any other genes. It has to be rearranged during development. It's from 1V, one variable regions, 1D diversity, 1J joining gene segment. There are many V, there are many D, and there are 6 Js in a human. So here, for example, you pick 1V, you pick 1D, you pick 1J, they rearrange it to become a productively rearranged antibody genes. This is then transcribed to give you RNA and give you proteins. So if you have a lot of variable region, a lot of diversity region, a lot of joining regions, that's how you can become around 1 billion different antibodies. So humanized mice basically is to replace the mouse antibody genes. So this is, let's say, the mouse rat, the mouse locus, with the human antibody variable genes. So you have a mouse where the heavy chains is part of it is from human, and then part of it here, the constant region is from mouse. You can do that also for the light chain. So you have both heavy chains and light chains, and this is called these humanized mice. And this was done through embryonic stem cell engineering. Takes a long time, but it has been done. So instead of producing antibody for spike proteins in mouse, and you get mouse antibody, if you inject this spike protein into these humanized mice, these mice actually have the constant region from mouse, but variable region from human. Then you can get S-specific B cell with a human mouse antibody chimera, basically. Right? Very interesting, right? So this is the whole variable region is already human. All you need to do is to replace the constant region with the human also. So then you isolate this B cell. So you just label them with, let's say, a spike protein that labeled with red fluorescence, and run it through a machines. We call it fluorescent activated cell sorter machines, FACS sorter. They were first invented at Stanford University. So you run this through a machines, and this machine has a laser that will detect the light, and the cell is then, has a charge that you just put a charge in there. So then the machine will say, okay, it's red. Let me just push it to one tube. Has other color, or let's say green, if you label it with green, but in this case, you don't even label it with green, just red. So just put red over here. So that's how you can actually separate B cell that's specific for spike versus other B cell, or versus other non-B cell. So you do this from a spleen of the humanized mice, or from this convalescent human. So then you take single B cell, you do a single cell RT-PCR. So that's reverse transcriptase PCR. So you make the cDNA basically from the RNA, and you isolate the variable region for heavy chain, variable region for light chain, which are human obviously, and then you fuse this in the molecular biology technique to constant regions from the human now, constant region human for the heavy chain and light chains, and then you put this DNA into cell. So, for example, Chinese hamster ovary cell, and the cell would then secrete a lot of antibody, and this antibody will be completely human. Okay. So then you have lots and lots of these cells that have, each of them have their own different antibody, and you can test individually for the ability to neutralize the virus. And you purify the antibody first, obviously. So this company actually have produced many different antibodies like this. There are around 200 of them, but about 9 of them actually have very high activity to neutralize SARS-CoV-2 virus. So over here is percent neutralization on the y-axis, and on the x-axis is the amount of antibody that you needed to neutralize 100% of the virus. So this is actually a soluble S2 protein. So remember, S2 is a receptor for the virus, but you can make soluble human S2 protein to overwhelm the system and block viral entry. And so this just doesn't work very well. You can see here you need a lot of the soluble S2 protein to block only 80%. But there are 9 different antibodies here that can block the viral entry very easily. So neutralize the virus. So this antibody in particular is very good. Even at 10 to the minus 10 molar here, you can block 100% of the virus. Okay, so I think they are in clinical trial. So that's a very promising drug. So let me just summarize quickly again this part. So in some COVID-19 patient, I told you cytokine storm can result in death, and you can use dexamethasone to help, even though it's not completely cure the patients, but decrease from 40% to 30% for those that's under ventilator. And for future very promising therapeutic drug, you can actually make antibody from the humanized mice and convalescent human using the FACS machines, and you just use molecular biology technique to produce the human anti-spike antibody in mammalian cell that's tested for the ability to neutralize the virus. And a cocktail of neutralizing antibody is currently in clinical trial, and really hope that this will work. Okay, so that's basically the end of my presentations, and I welcome any questions. So thank you for listening.
Okay, thank you very much, Sudah. Thank you very much, Prof. Astar, from incredible lecture. Frankly speaking, my brain is not really enough for digest all. But so before I need your and Professor Iris as panelists, may I raise an issue? Yeah? Number one. So according to you, the researchers and Professor Iris as a clinician, in this third world war, human against COVID-19. Who will win? So you know that we already many times against viral diseases like SARS, like swine, whatever. But why they are not spreading widely worldwide like a COVID-19? Why the character is different, right? We couple of times, right? Like had an outbreak, but it's not a pandemic. So in my life, this is the first pandemic I have ever had. And why? Is it because the character of COVID-19 is different? Is it because they produce so quick and why so quick and so fast? And why again they spread so quick? Why swine flu? Why bird flu not spreading that worldwide? And again, so finally, we will find the vaccine against the virus, or finally, the nature will select. For instance, so probably that we expect that COVID-19 someday probably will be weaker, just like a common cold everywhere, but they never kill us at this much. So this is the first issue. So again, in this third world war, who will win and how to win? So again, and after that, again, the character of the COVID-19, why so different with others? And again, after that, Professor Astar, do you think that COVID-19 is really coming from the laboratory because of while they're doing experiences and then I don't know how, or because this is because of the, I don't know, because the word changed too much, because the human make every change, then the virus also changed their body into resistancy as well. Which one you believe? Is it from the laboratory or this is the natural processes and caused the pandemic? So the first, Prof. Iris and Prof. Astar, can you please enlighten us? So who will win?
Okay.
Please.
Okay, Prof. Ika. You know, I just want to address the first questions. So I think the differences with this virus that make it very deadly and pandemic is because there are many humans that have no symptom. So OTK, OTK, orang yang tidak ada gejala. And that's the biggest problem. And swine flu, the human-to-human transmission is very, very low. But the human-to-human transmission is very high. So that's already one difference. SARS-1, the initial SARS, or MERS, when people get virus, they get sick. So they stay home and they sleep, you know, they sleep and stay home and they don't infect other people, other maybe their own family members. But for this virus, the one that's really, really bad is the fact that there are a lot of asymptomatic people which spread the virus very quickly. Okay, so that's the main reasons. And instead of getting weaker, they actually get stronger. So there is this D640G mutations that has become a dominant virus. You know, initially it has aspartic acid in one of the positions. It becomes glycines. And that mutation actually make it more easily transmitted from human to human, and they replicate better. So let's not hope that, well, I hope you'll get bigger, but unfortunately, I don't think that's going to happen. So the only way to combat it is using vaccines and obviously using antibody drug that is almost like passive vaccine. So that's like almost like before you get the vaccines. Is that the question you have? And then the third question is whether it's lab-made or human or nature-made. Well, all the evidence so far is actually nature-made. It's from bat, from Palalawa, because it has 96% homologies, and maybe go through this pangolin trungeling as an intermediate species. There is a lot of evidence I don't have to go through it here. So it shows that it's not lab made, that it has to go through immune evolution. For example, the spike protein has many modifications that show that it has tried to evade the immune system. So you cannot make it in the lab. It has to go through an organism. So that's one reason.
Actually, in your lab, can you also modify virus to be another model of virus in your lab?
Well, you can modify the virus, sure. But, you don't try to make a virus like this.
Okay, thank you very much. Thank you very much. So, Prof. Iris, what is your opinion, Prof. Iris?
Yeah, I think I agree with the Professor Astar. The answer is already very clear, yeah. But I want to add maybe our system is not yet known about the virus. This is a new virus. That's why we don't have any sensitization before, yeah. And then the immune system is going to make antibody, but cannot so fast, yeah, because the virus is very we call very dangerous, yeah, dangerous, and maybe like a super antigen. I don't know it is like a super antigen or not, yeah, because until now we still don't know about the virus like that, yeah, like this. I agree with Professor Astar. But Professor Eka, we have many questions. I don't think we have enough time to answer all the questions. But if you don't mind, maybe we can move on to the question.
Yes, go ahead. And maybe before that, one question from Professor Ari, Dean of UPI. So how about rapid tests in the hospital? Is it useful to screen the patient for surgery forever? Can you please answer? Thank you very much. Go ahead.
Yeah, please, Professor Astar.
Oh, I think the only good test is PCR test, unfortunately. It has to be done very quickly also. The rapid test, that's an antibody test, is only telling you, what happened to the body. It's obviously the body will have IgM, will have IgG, but that really doesn't tell you what happened to the virus. The virus, if it's negative, you can still have virus. But that's the problem. When you say negative, a lot of people say, "Oh, I'm negative." Wow. So so they say, "Great, you can go home, you can do all kind of stuff," but it's not true. Asymptomatic patients and patients who are in the early symptom, they have a lot of virus, but they don't have the antibody yet. They will be negative in the rapid test.
Yeah.
For surgery, I would really like to see the PCR test.
For the surgery, how do you think, Professor Astar, we need to have not PCR, but a PCR test? Do you think we need to do that?
Yeah, perlu sekali PCR test.
Because it's very expensive, yeah.
Yeah, it's expensive, but if you want to be safe, you really the doctor wants to be safe, right? You don't want the doctor to get virus. That's the only way to do it.
It's not rapid test, yeah. The answer is PCR.
Yeah, there is also a rapid test for antigens, but I don't think they are that. I don't know if they have it in Indonesia. China actually have rapid test the antigen, but they're also not very sensitive. It's fast. The other way to do it is, the Abbott lab. It's also expensive. There's some machines made by Abbott lab. That's PCR, and you can get the result within half an hour. Donald Trump, he doesn't use mask and anything, but he get tested all the time, and anybody who going to the White House are being tested, and they use the Abbott lab test. It's very fast, but it's also expensive.
Oh, very good. So maybe Prof. Ari and Prof. Budu, you should inform Minister that Abbott is very quick and very important, instead of the other waiting for a couple of days to under the PCR test. Okay, Prof. Iris, go ahead with your question.
Okay. Another question. I will ask Professor Astar. This is a question from Dr. Farid Kurniawan. In older adult and patient in comorbid, ini campur-campur bahasanya ya, it's okay. Ada older adults and pasien dengan comorbid, diabetes, hypertension, and obesity, gejala COVID-19 relatif lebih berat dan mortalitas lebih tinggi. Apakah peran spesifik from the response imun berbeda pada orang-orang pasien tersebut? Kenapa berbeda? And ada patofisiologi yang lain.
Itu tapi agak susah juga. Nah, jadi saya lanjutkan ya Bapak Ibu. Bagaimana pelaksanaannya untuk project ini?
Ada
Jadi
suara.
nanti kita mengikuti sampai kita arahkan dulu.
Halo?
Dr. Stephen, tolong di-unmute yang lain.
Ya.
Ya.
Maksudnya apa ada bedanya immune system di antara banyak comorbidity itu ya?
Yes.
Selama ini kita tidak ada buktinya kalau ada bedanya. Jadi mereka hampir sama terus. Belum ada studi yang bisa membedakan immune sistemnya orang yang ada cardiovascular problem atau ada diabetes atau obesity. Semuanya ada hyperinflammations. Tapi hyperinflammation is relative ya, tidak bisa persis berapa nilainya gitu dan bedanya sama semua.
Yeah. And then the other question, receptor ACE2 ada 81, 82, and 84. Apakah clue virus COVID-19 masuk ke 81, terjadi vasoconstriction production, ROS, and then 82 terjadi vasodilatation. Bila masuk ke 84, mengaktifasi PAY1 ya, PAY1. What do you think?
S2 81 itu apa itu? S2 hanya satu.
S2 reseptor dibagi ada 81, 82, 84 like that?
Enggak lah. S2 hanya satu. Nah, ada polymorphismnya, sama terus.
Oke. Jawabannya is the same ya, Dr. Rizwanto. Enggak bagiannya, jadi semuanya only H2. Yeah. And then from Dr. Ravenal Padang, mau tanya kenapa variasi sekali respons imun dalam memproduksi antibody, IgM, CD4, CD8 terhadap SARS-CoV-2. Is there any factor yang mempengaruhi respons imun tersebut?
Mengapa variasinya banyak antara different patient ya?
Ya.
Yang kita nggak tahu, memang tubuhnya manusia agak lain-lain ya. Jadi ada mungkin yang imun sistemnya kuat, ada yang imun sistem yang tidak kuat. Tapi ada satu lagi interesting fact. Common cold itu ada 4 common cold ya coronavirus. Ya, ada studi yang lihat T-cell untuk SARS-CoV-2. They found that orang yang nggak pernah dapat COVID-19, tapi ada T-cell-nya untuk SARS-CoV-2. Mengapa? Karena dia dulunya kena common cold ini. Jadi some of the epitope, some of the peptide yang dari common cold itu bisa produce T-cell specific for the SARS-CoV-2. So ini hanya spekulasi. Mungkin individual itu karena udah ada CD4 helper T-cell dari common cold yang bisa recognize SARS-CoV-2, jadi B-cell-nya production lebih cepat. Nah, ya itu aja spekulasi, tapi memang ada data kalau ada CD4 T-cell di orang yang tidak pernah kena COVID-19, sama sekali tidak pernah kena, tapi ada CD4 T-cell yang bisa cross spesifik untuk.
Yeah. Okay. From Dr. Melania, consideration response to COVID-2 is worse in patient with defected immunology response. Can we identify specific cells that actually responsible for this response?
Gimana?
Considering response to COVID-2 is worse in patient with defected immunology response, can we identify specific cells that actually respond? Ya, saya ulang ya. Considering response to COV-2 is worse in patient with defective immunology response, can we identify specific cells that actually responsible for this response? Pertanyaan dari Dr. Melania. Jadi,
maksudnya orangnya ini udah tidak ada immune sistemnya atau gimana?
Ya, jadi yang karena COV-2-nya dia getting worse ya, imunologi responsnya. Apakah kita bisa identifikasi sel apa yang untuk respons ini? Kenapa dia menjadi lebih jelek? Ada enggak sel-sel imunologi yang menjadi lebih fokus kenapa dia jadi jelek? Sel-sel mana kira-kira? Can we identify specific cell? Sel-sel imun system.
Sel-sel imun system ya itu B cell, T cell. Ya, kalau mau identify ya itu aja. Jadi B cell, CD4 dan CD8 T cell.
Hanya
itu. Yang lain sama semua. Anyway.
Check CD4 or CD8 ya to know the immune system.
CD4, CD8. Yang lainnya semua sama anyway. Yang innate immune cell itu tidak berubah ya.
It's the same action ya. It's just the same. CD4 and CD8 ya. If you want to check in the laboratory. Yeah. Okay. And then from Bana, Prof. Astar, how about applying vaccine through the skin? As dendritic cells are found in the skin, thus the antigen of the COVID-19 will be bring to lymphatic node and activate the T cell without activating the ACE2 receptor. Jadi pertanyaannya kalau ada vaksin masuk ke melalui kulit dan ada dendritic cell di kulit itu, bagaimana vaksinnya bekerja tanpa mengganggu ACE2 reseptor.
Vaksinnya tidak perlu kerja lewat ACE2. Tapi kalau kamu injek ke skin, ya muscular, kalau ke muscle ya, itu langsung di-deliver ke lymph node. Itu jalannya gitu. Jadi vaksin diinjeksikan ke kulit atau ke muscle ya.
Muscle,
itu semuanya masuk ke lymph node dulu. Nah, di lymph node itu bisa activate dendritic cell.
Ya, jadi tidak melalui ACE2 receptor, ya langsung ke lymph node.
ACE2 kan hanya untuk kalau virusnya ada. Ini vaksinnya enggak pakai virus. Virusnya virus adenovirus 5 itu common cold, enggak pakai ACE2. Atau yang dari Moderna itu pakai mRNA ya. mRNA jadi tidak ada virus sama sekali. Ada yang DNA vaksin, ada yang RNA vaksin, itu tidak pakai virus.
Ya, vaksinnya vaksin mati ya, kita vaksin mati, jadi bukan vaksin hidup.
Oh, kalau vaksin yang misalnya dari Sinovac itu ya, itu vaksin yang inactivated ya. Kalau itu ya also has to go to, that's true. Itu ya has to go through itu lymph node juga. Dan somehow harus masuk ke immune cell tidak lewat ACE2, sebabnya immune cell tidak ada ACE2.
Ya.
Jadi harus masuk pakai karena pakai phagocytosis kan dendritic cell macrophages bisa phagocytosis. Jadi kelihatan virus yang sudah mati, jadi bisa di engulf, jadi engulf dari sana bisa present ke T cell. Lalu bisa activate T cells.
Jadi berbeda cara kerjanya ya.
Ya, tergantung vaksin apa. Ya, beda sekali kerjanya. Kalau inactivated virus ya harus pakai ACE2 sebab immune cell ada ACE2 ini. Tidak lewat paru-paru toh, kalau lewat kulit.
Ya, jadi karena kita IM intramuscular and then straight to the lymph node ya. Sekarang pertanyaan berikut dari Christopher Hans. Wouldn't using corticosteroid secara teori bagaimana bisa meng-clearance viral from the patient? About the corticosteroid dexamethasone, like that. Bagaimana?
Sorry saya agak jelas ini koneksinya. Gimana lagi? Coba ulang ya.
Pemakaian dexamethasone.
Oh, dexamethasone.
To slow down the viral clearance. Bagaimana cara kerjanya?
Oh, dexamethasone is a non-specific steroid, glucocorticoid, just anti-inflammation in general.
Ya, maybe
udah sering pakai dari dulu.
Bukan untuk viralnya, tetapi mungkin for the inflammation ya.
Ya, bukan virusnya, inflammations.
lebih cocok cytokine storm mungkin ya, Prof. Astar ya.
Ya, memang hanya bisa untuk cytokine storm.
Not for the virus.
Oh, benar. Jadi kalau early jangan pakai dexamethasone, hanya untuk severe patients.
Ya, itu
Halo,
Prof. Eka.
Boleh satu pertanyaan selingan yang rada silly question. Prof. Astar, so how is situation in the US now? What I heard is, dulu kan orang nggak mau pakai masker, mau-maunya saja, dulu very hard. Tapi is it better now? No, my question is, sebenarnya kita itu perlu apa tidak sih rapid test di mana-mana? Apakah ini karena orang panik saja? Apakah ini karena advice dari WHO aja? Buat apa di rapid test? Kan ujung-ujungnya sebenarnya adalah tolong dijaga perilakunya. Pakai masker, social distancing, cuci tangan, sangat mudah. Misalnya orang Jepang kan hidupnya rapi, bagus, apik, itu nggak perlu rapid test juga bagus-bagus aja misalnya. Katanya begitu kan, di Jepang itu rapid testnya nggak banyak, tetapi juga kematian juga nggak banyak. Jadi sebenarnya kita itu bodoh apa enggak sih buang-buang duit sekian ribu triliun untuk rapid test? Ujung-ujungnya kan number one is pokoknya hati-hati, jangan sampai ketularan, jangan sampai menularkan. Jadi my silly question, Prof. Astar, siapa yang punya ide rapid test? Sebenarnya kenapa kita harus pakai rapid test? Gara-gara kita panik, kita nggak ngerti kan, begitu kena attack semua bingung. WHO ngomong A ikut A, B ikut B. Is it too silly for you, Profesor Astar?
Ya, saya tahu. Ya di US ini memang orangnya bandel-bandel semua, nggak mau pakai masker. Nah, karena itu virusnya gila-gilaan sekarang di sini, unfortunately. Tentang rapid test ya, kalau menurut saya it's a waste of time memang. Nggak perlu pakai rapid test. Di US nggak ada yang pakai rapid test. Kalau sakit, you do PCR. Jadi social distancing, wash your hand, cuci tangan, pakai masker, ya itu paling penting. Kalau sakit ya PCR test, you have to do PCR test. Tapi PCR testnya harus kembali cepat, dalam 1 hari 2 hari. Kalau 5 hari 7 hari ya udah terlambat, apa gunanya waktu itu ya. Rapid test sih nggak perlu, rapid test antibody kerja apa? Aku tahu, bakalnya pasti ada antibody. Dan kalau kamu ada IgG, berarti mungkin virusnya udah habis. IgG positif, your immune system is very good, and the virus might be gone, udah hilang. Then it's great, kamu immune. Hanya itu aja. Jadi kalau rapid test, menurut saya di seluruh dunia jarang dipakai. Indonesia yang sering pakai, aku juga nggak begitu ngerti. Dari permulaan saya udah nggak setuju, udah pernah bilang dengan.
Nah, justru itu banyak orang. Makanya yang saya sejujurnya, saya itu mungkin bodoh sekali kok tanya begitu. Ini tuh siapa yang bermain? Apakah gara-gara WHO-nya bingung? Semua ikut bingung. Kembali lagi kan begitu, kita buang duit berapa dan ujung-ujungnya ya tadi itu kan buat apa? Nah, mungkin nanti Prof. Iris ya sebagai ketua Ikatan Immunology mengatakan Pak Jokowi kalau perlu buat apa kita buang rapid test? Pak Joni barangkali di Surabaya bagaimana Pak Joni Wayuhadi itu. Di sana di rapid test apa gunanya? Kira-kira kan ujungnya juga sama saja ya. Jadi gimana kalau setuju kita segera usulkan aja ke Pak Jokowi. Kita menghabiskan berapa ribu triliun duit hanya untuk beli rapid test. Dan ujungnya kan jujur hanya saya setuju dengan Prof. Astar tadi, orang Amerika mungkin gila, tapi mungkin juga benar. Dalam artian ngapain di rapid test? Jadi kita sekarang orang heboh, semua jadi bisnis besar.
Oh
jadi,
ya jangan.
Jadi bisnis opportunity yang besar, tapi yang rugi ya nggak tahu. Ya jadi Dr. Joni boleh nggak komentar sedikit, dikasih sedikit Dr. Joni Wayuhadi. Apa yang terjadi di Surabaya? Karena ini bosnya Surabaya. Perlu nggak rapid test? Stop aja duitnya buat beli PCR atau buat beli
bikin PCR supaya lebih murah. Itu yang paling penting.
Ya, Gimana Pak Joni, perlu nggak rapid test? Barangkali kita harus ngapain di rapid test? Masih mute itu. Pak Joni?
Sudah di-unmute Prof, tapi mungkin mic-nya Pak Joni. Sudah di-unmute.
Pak Joni? Pak Joni? Dr. Joni? Hah? Join audio Pak Joni. Pak Joni? Audio. Pak Joni?
Audionya belum nyala, Dok. Dr. Joni.
Pak Joni? Audio. Pak Joni audio.
Audio di sebelah kiri. Ada mute, unmute.
Pak Joni?
Sudah kita unmute, mungkin koneksi audionya bermasalah, Prof. Kemungkinan besar, Prof.
Ya.
Oke, nanti kalau gitu saya telepon beliau dulu aja ya sebentar. Barangkali Prof. Budu dan Prof. Ari bagaimana pendapatnya? Perlu nggak sih sebenarnya?
Kalau saya, halo.
Halo, ya Prof.
Halo.
Budu.
Ya, silakan Prof. Budu.
Prof. Eka, bukan masalah apa bisa kita buat something question untuk Prof. Astar.
Silakan.
Yeah. The first question is, for example, misalnya saya sendiri, ini kan baru recovery from COVID-19.
Waduh.
Ya, jadi tadi waktu Prof. Astar presentasi, I saw that the immunoglobulin G will be maybe
Terus ya.
take long time for 3 months or more. Jadi apakah saya sendiri ini sudah bisa saya anggap it is safe for maybe 1 month or 2 months? No need to in protokol kesehatan begitu. Kemudian yang kedua, yang the second, Prof, I'm sorry. The second itu adalah itu dulu, first question dulu, baru nanti saya ini lagi yang kedua.
Ya, ada IgG, jadi saya assume IgG-nya bisa neutralize virus, dan at the same time saya juga kira pasti ada CD8 positive killer T cell. Jadi bisa assume kalau Prof. Dokter sana bisa imun lah, at least beberapa bulan. Nanti turunnya ini agak kontroversial, ada orang di New York, study New York bilang antibodinya titernya sama terus sampai 3 bulan. Ada satu lagi grup katanya agak turun, tapi turun belum, tapi mereka tidak lihat cytotoxic T cell. So either way, paling sedikit imun tebal 3 bulan, 4 bulan pasti itu. Saya kira.
Oh, artinya
tahu saya nggak tahu.
Tapi itu bertahan agak lama, kira-kira 4 bulan begitu ya?
Ya, sudah itu saya tidak tahu ya. Dari 4 bulan saya kira nggak apa-apa.
And the second from this may be very important questions because I saw there is some like ICE receptor for COVID-19. Dan ICE receptor is related to also about the blood pressure and so on.
Ya.
Saya ini tidak pernah hypertension, but waktu saya COVID-19 2 weeks ago, itu the second day and the 7th day, 1 week after, tiba-tiba saya pergi ke rumah sakit karena suddenly my blood pressure is high, guys. High. Jadi saya ke rumah sakit bukan karena batuk, karena demam, tapi saya punya tekanan darah tiba-tiba naik.
Wow.
That is relation to COVID-19?
Ya mungkin cytokine storm-nya ada sedikit, atau juga memang virusnya juga bisa affect vascular ya, bisa S2 ada di vascular juga. Jadi bisa juga karena itu. Jadi S2 turun, lalu blood pressure naik.
Yeah.
Because IC is inhibitor for
OS, ya.
ya, okay,
Ada studi, kalau berdasarkan baca studi juga gitu, orang yang hypertension yang makan ACE inhibitor, ada yang stop, ada yang kena COVID-19 lalu stop, ada yang kena COVID-19 diteruskan. Lalu kita lihat mortality-nya gimana. Jelasnya mortality-nya lebih rendah kalau nggak stop. Jadi ACE inhibitor harus diteruskan.
Oke.
Terima kasih ya. Halo.
Thank you, Prof.
Ya, ya.
Thank you, Prof. Eka.
Ya, Prof. Eka.
Pak Joni Wahyuhadi. Pak Joni bisa. Pak Joni belum bersuara.
Wih, bagus sekali ini. Boleh jadi bukan sebenarnya kemarin.
Pak Joni belum bisa, Pak Joni.
Belum bisa.
Pak Joni mic-nya belum bisa. Pak Joni mic-nya belum bisa, mic. Pak Joni mic-nya belum bisa. Pak Joni mic-nya belum bisa. Belum bisa Pak Joni mic-nya. Barang ini Prof. Ari dulu, Prof. Ari. Prof. Ari ada? Prof. Ari masih ada? Halo Prof. Ari. Halo Prof. Ari masih ada?
Prof. Ari kelihatannya sudah ada pertemuan lain, Prof.
Oke.
Sudah ada pertemuan lain, Prof.
Jadi dari Prof. Iris lagi masih ada, silakan Prof. Iris.
Pertanyaan ini, ya oke. Pertanyaannya dari Dr. Ai Salim. Apakah perlu rapid test for follow-up patient melihat reaksi kekebalan pada patient? Karena mengingat pasien di pedalaman atau di kota kecil atau di kampung sulit dilakukan PCR. Boleh nggak di kota kecil untuk maintain pasien melihat kondisinya udah baik atau belum pakai rapid test? Bagaimana Prof. Ashar? Ini kota kecil, nggak ada PCR.
Ya. Kalau menurut WHO, kan sekarang guideline-nya kalau udah nggak ada simptom 3 hari, dan udah lewat 10 hari, jadi 13 hari, udah nggak infectious. Jadi saya kira kalau nggak ada PCR, ya nggak perlu PCR udah. Jadi kalau udah lewat 13 hari, ya nggak apa-apa udah. Nggak perlu infectious. Rapid test juga nggak perlu. Pasti IgG positif lah.
Ya. Nah. Jadi tidak perlu untuk follow up ya. No need for follow up patient.
Ya.
And then satu lagi dari Dr. Gatot Surabaya. I think SARS-CoV-2 can also be recognized by immune system through interaction with TLR2, TLR3, and TLR toll-like receptor 7. Not only through ACE2 receptor, which is used by the virus to infect host cells. Bagaimana pendapat Prof. Ashar? Prof. Astar, ada toll-like receptor 2, toll-like receptor 3, and toll-like receptor 7. Jadi not only ACE2 receptor, ada reseptor yang lain.
So, ACE2 itu adalah reseptor yang virus ini pakai untuk masuk sel, infect sel. Lalu toll-like receptor TLR3, 7, or 9 itu adalah innate immune cell receptor. Jadi ini mereka recognize virus in general. Jadi TLR3 recognize double-stranded RNA, TLR9 recognize the CpG DNA, lalu TLR7 recognize single-stranded RNA. Jadi it's a response. Jadi part of the TLR receptor may be responsible untuk memberikan cytokine, inflammatory cytokine dari infected cell. Jadi really is not the receptor untuk virusnya. It's a general innate immune cell receptor.
Ya. Jadi COVID-19 ya, only H2 ya, Profesor Astar.
TLR2 bukan untuk virus. Virus is TLR3, 7, and 9. Tapi juga ada what do you call it? Pattern recognition receptor yang RIG-I, namanya RIG-I juga ada. Lalu NAP-R3, lots of them actually, banyak sekali.
Yeah, but that's only for the SARS-CoV-2 is only H2 receptor.
Ya, yang lainnya itu hanya is immune response.
Okay, jelas ya. And then from Dr. Suryani Makassar, apakah autofagi yang merupakan salah satu mekanisme imun kita dimanfaatkan oleh COVID-19 for replication? Autofagi.
Setahu saya enggak. Autophagy itu hanya remove, unwanted organelle, enggak dipakai dengan virus ini.
Ya. And then ada lagi fungsi dari sel like T regulator, macrophage, and mediator yang bekerja sebagai anti-inflammation like IL-10 mungkin mengalami anomali respons atau berespons tidak cukup adekuat. Apakah sudah ada studi yang telah explore about fungsi anti-inflammation IL-10?
Ya, IL-10 juga dikeluarkan dengan banyak pasien ya. IL-10 is suppressive cytokine. Tapi in this case saya nggak tahu ada studi tentang IL-10.
Nggak ada ya studi ya.
Bulan ini ya. Papernya banyak sekali, tapi yang IL-10 masih jarang.
Tapi IL-10 kan actually is itu ya menghambat inflammation.
Ya, suppressive.
Very good. Ya, jadi ada keseimbangan between the Nah, kemudian ada pertanyaan lagi dari yang lain. When IgG response juga tidak berbeda antara yang sembuh dengan yang interinflammation. The question is, apakah subtype IgG yang ada mengalami dominasi subtype IgG yang berbeda? Jadi subtype IgG ada IgG1, IgG2 yang saya tahu ya sampai 4 ya. Apakah ada berbeda pada SARS-CoV? Respons?
Nggak tahu ada beda nggak. Nggak ada paper yang bilang kalau beda. Semua orang lihat IgG aja, nggak di subtype aja.
Ya.
Belum di subtype.
Jadi secara umum ya, Prof. Astar ya, IgG.
Dan bedanya IgG 1, 2, 3, 4 itu fungsinya hampir sama. Jadi meskipun berbeda ya, maksudnya apa nggak tahu ya.
Ya.
Karena itu nggak ada yang ngurus.
Ya, oke. And then from Dr. Stephen.
Ya, Prof. Astar, Prof. Iris, kalau boleh bertanya, there is several question from autoimmune patient yang mungkin saya mau minta pendapat dari Prof. Iris dan Prof. Astar mengenai vaksin. Jadi kalau kita lihat beberapa vaksin yang sedang di-develop itu kan ada yang dengan conjugated, ada juga yang inactivated, kemudian ada juga vaksin-vaksin yang dari material dari RNA virus itu sendiri. Nah, kita tahu kalau pasien dengan autoimmunity di dalam tubuhnya memang sudah terjadi hiperinflamasi. Tetapi di sisi lain, respons pembentukan antibody terhadap vaksin juga berkurang. Nah, kira-kira bagaimana menyikapinya pada saat kita memerlukan vaksinasi untuk pasien-pasien yang memang sudah punya reaksi hiperinflamasi di dalam tubuhnya, khususnya adalah untuk pasien-pasien dengan autoimunitas, Prof. Karena mereka kan salah satu populasi yang berisiko tinggi. Terima kasih, Prof. Mungkin Prof. Iris dan Prof. Astar bisa berkolaborasi memberikan jawaban.
Ya, silakan Prof. Astar.
Autoimmune itu biasanya very specific. Misalnya autoimmune untuk multiple sclerosis, itu autoimmune untuk myelodysplastic protein ya. Jadi in general immune sistemnya seharusnya is okay, bukan hyperinflammation. Ya tergantung phase 3 gimana ya. Hopefully it's okay. Tergantung vaksin gimana yang phase 3 bagus. Menurut saya sih tidak perlu terlalu mikir terlalu banyak-banyak. Sebenarnya autoimmune disease itu lain-lain. Tergantung disease apa dan bukan general hyperinflammation. It's very specific T cell untuk specific antigens.
Ya, betul, Dr. Stephen. Jadi for autoimmune disease, we must be careful to give the vaccine ya, because autoimmune disease kita tahu the system immune is antibodinya menyerang tubuh kita sendiri. Jadi endpoint from the vaccine is also antibody. That's why we must be careful to give the vaccine to the autoimmune patient. Ya. Kalau vaksin hidup udah pasti nggak boleh, tapi vaksin mati itu kita lihat nanti ya inactivated vaccine according to the patient. If the patient still take a immunosuppressant or a corticosteroid in high dose, we cannot give the vaccine because it will be no response antibody because suppressed from the from the obat-obatan ya, from the therapy ya, from the immunosuppressant.
Ya Prof, juga kan salah satu vaksin yang sedang diuji coba itu yang dari Sinovac itu kan menggunakan aluminium sebagai adjuvan Prof. Ya. Dan kita tahu itu bagaimana Prof? Apa pendapat Prof mengenai penggunaan adjuvan seperti aluminium Prof? Terima kasih.
Ya, memang adjuvan aluminium kita tidak anjurkan untuk orang autoimun ya, karena dia sebagai semacam kayak bukan logam ya, tapi semacam begitu nggak bisa meninjus autoimunnya sendiri. Jadi kita juga harus melihat justru banyak vaksin yang membuat autoimunnya di flare up because the adjuvan, ya not because the inti dari vaksinnya sendiri, but because the adjuvan. Justru itu harus hati-hati sekali dalam pemberiannya. Betul, Dr. Stephen. Ya, Dr. Stephen.
Ya, Prof Eka, Prof Iris, Prof Astar, apakah ada lagi yang ingin disampaikan, Prof?
Pertanyaannya sih masih banyak belum terjawab, tapi kalau waktunya habis, mungkin kita bisa kirim ke Prof Astar.
Kita masih ada waktu kurang lebih sekitar 8 menit lagi, Prof, sebelum jam 10.
Saya mau bicara lagi dengan Prof Ika. Itu anjurannya Prof Ika betul sekali. Jangan pakai rapid test. Uangnya dianjurkan ke PCR test. Sungguh. Itu sungguh. Menurut saya itu paling penting. PCR test memang mahal, tapi kalau lebih banyak orang mungkin bisa dimurahkan ya, nggak tahu ya. Tapi rapid test memang gunanya lebih banyak. Jadi daripada buang uang ke rapid test, lebih baik ke PCR test semua. Nah, kalau bisa, ya pemerintah bisa nanggung harganya, itu paling bagus. Di US ini PCR testnya semua free memang. Kalau nggak free memang susah sih. Yang mahal sekali, kalau komersial ada berapa juta untuk tes. Ya nggak mau memang biasanya. Ya memang harus gitu. Memang ya apa ya? Memang mahal, tapi itu penting untuk membasmi virus ini.
Satu pertanyaan, Prof. Ashar menyambung rapid test. Untuk epidemiologi, apakah kita masih bagus pakai rapid test just for epidemiology, only for knowing somebody already contact with the virus?
Kalau bisa lebih baik pakai ELISA.
ELISA ya, oke.
Lebih spesifik dan ELISA juga murah, bukan mahal. Bisa.
ELISA is a rapid test or
ini? Bukan rapid test, harus pakai mesin tuh, harus pakai mesin.
Oke, berarti metodenya berbeda ya.
Metodenya berbeda. Very specific dan juga tidak begitu mahal. So, I think lebih baik itu. Sebabnya rapid test itu masih kurang spesifik ya. Ya kalau coba ELISA dulu menurut saya.
Ya, jadi pemasukannya baik-baik ya pada diskusi kita. Jadi tetap kita pakai PCR ya, not rapid test. Jadi itu kesimpulan yang baru kita bisa ambil pada banyak diskusi pertanyaan-pertanyaan yang membingungkan kita semua selama ini. Tapi Prof. Asar sudah menjawab dengan tegas bahwa rapid test itu tidak banyak memberi informasi bahwa orang itu sakit atau tidak, karena hanya menyatakan bahwa orang itu sudah terkontak, tetapi IgG ya berarti dia sudah lewat, lewat. Itu. But one more question, Dr. Asarnya bisa membantu semuanya. Apakah bisa reinfection? Because the antibody alami, ya, the natural antibody kita bisa terbentuk after we get the infection. But maybe only 30% or 40% like that. That means we can get reinfection again. Is there any
Ya, katanya ada anecdotal evidence kalau ada orang yang bisa kena infections. Tapi seharusnya ya enggak ya. Most of the time seharusnya enggak kena infections. Tapi memang ada, kita masih belum tahu semuanya ya. Enggak tahu antibody titernya gimana, lalu cytotoxicity-nya gimana, sudah berapa bulan ya. So, paling kalau mau hati-hati ya, hati-hati aja supaya enggak kena ini. Ya, kamu assume aja bisa reinfection lebih baik.
Ya baik, silakan Prof Eka. Ya silakan Prof.
Iya nggak, Dr. Joni?
Iya bisa dengar saya.
Sorry, ada trouble. Saya penting, beliau itu panglimanya dari Jawa Timur tentang gugus tugas. Gimana saat Dr. Joni perlu nggak rapid test? Tolong lewat HP.
Ya, jadi sudah dengar ya, bisa lewat HP ya. Nah, jadi untuk di Jawa Timur sesuai dengan pedoman yang baru, kita tidak lagi melakukan tes. Memang pada awalnya ya karena memang kebijakan pemerintah tes itu sudah terlanjur dibuat ratusan ribu. Nah, sekarang sudah tidak dipakai itu tes. Screening kita memakai PCR juga sudah cukup banyak. Kemudian untuk repeat test itu dipakai untuk surveillance. Jadi surveillance di rumah sakit. Tidak lagi pakai repeat test.
Ya, oke. Terima kasih ya. Terima kasih. Ini panglimanya. Jadi barangkali ada pemirsa saat ini yang cukup dekat dengan Pak Jokowi. Siapa yang berani bisikin jangan beli lagi, ngabisin duit negara. Nanti kalau enggak percaya saya teleponkan Prof Astar. Ini maaf ya, ini ide bodoh ya. Barangkali Prof Iris kenal juga Pak Jokowi ya. Ini enggak perlu lagi PCR, buang duit. Kasihan yang bayar di negara dan tidak gratis di sini Prof Astar ya. Jadi kembali lagi
PCR penting.
Mendingan PCR ya.
Ya, mendingan PCR.
Oke ya, setuju. Nanti kalau Pak Jokowi tanya, aku suruh telepon Pak Astar ya.
Ya, pasti.
Atau Pak Joni bisa tolong Pak Menteri Kesehatan kan lagi di Surabaya.
Oh ya.
Jangan, tolong besok dibisikin. Serius menurut saya ini problem ya. Kita nggak tahu saat ini. Kita buang duit gitu banyak ya.
Iya.
Baik, terima kasih Dr. Stephen. Barangkali ada yang lain nggak?
Nah, aku bilang lagi, bukan hanya kehabisan uang, bukan hanya buang-buang uang untuk rapid test, tapi kalau negatif masih mungkin infectious. Nah, itu yang takut.
Ya, jadi dulu itu idenya siapa toh, Prof Astar? Rapid test itu dari Amerika atau dari WHO?
Bukan dari Amerika, saya tahu. Di Amerika nggak pakai rapid test.
Nah, siapa dulu yang melontarkan di dunia?
Oh my God.
China. Tapi China juga nggak pakai. Rapid test harusnya untuk surveillance.
Pertanyaannya dulu siapa yang menghebohkan? Apakah ini proyeknya WHO atau bagaimana? Saya nggak ngerti kok sampai semua
bukan WHO. Saya dulu dengar Indonesia mau ke rapid test, saya udah bilang, waduh, gimana ini?
Harusnya
mau channel kanan kiri.
Ya, jadi maaf ya, ternyata kita, ya sorry ya, ternyata saya bodoh, ternyata lumayan ya. Jadi nanti kita rame-rame ngomong pemerintah, jangan beli lagi rapid test gitu ya.
Ya.
Baik, silakan Prof Iris masih ada lagi, atau Dr. Stephen masih ada lagi enggak? Sebelum kita akhiri, ada post test oleh Dr. Stephen.
Ya, jadi kalau cuman untuk ini saja menegaskan, jadi kalau untuk orangnya sakit atau tidak itu kita pakainya PCR, bukan rapid test. Tapi mungkin untuk surveillance saja mungkin ya, Prof Asar ya. Kalau surveillance epidemiologi,
ya, surveillance bagus ya.
Kita bisa pakai karena hanya untuk melihat berapa orang yang sudah terpapar atau belum, ya kan seperti itu ya. Maybe
orang yang asimptomatik pakai surveillance, pakai antibody.
Ya, only for
bulan setidaknya masih ada.
Ya, but not for diagnosis ya, not for diagnosis. Oke.
Untuk kita hanya untuk epidemiologis dan untuk mathematician saja ya.
Ya.
Rapid test untuk human being use ya. Oke, thank you very much. Ini saya rasa important sekali dan aku nggak berani ngomong, ternyata Prof Astar yang berani ngomong.
Ya.
Saya akan teruskan ke Pak Doni Monardo Sana kalau perlu kita teruskan ke Pak Menteri supaya ini dipertimbangkan, jangan diri lagi, tidak ada gunanya ya.
Ya.
Oke, saya rasa udah jam 10 ya. Dr. Stephen, terima kasih. Prof. Astar, thank you so much.
Oke. Ya, thank you. Ya.
Dan
Oke.
kita
Baik, Prof.
Oke, mari.
Izin untuk menutup acara ini, Prof. Iris, Prof. Astar, Prof. Eka, Prof. Ari, Prof. Budu, terima kasih atas kehadirannya, Dr. Joni, atas partisipasinya di dalam acara ini. Izinkan saya juga untuk mengingatkan bahwa untuk mendapatkan e-certificate bisa mengisi form di bit.ly/form UPH Berkeley. Dan untuk medical students ada post test di bit.ly tes UPH Berkeley. Jadi jangan lupa ini huruf besar dan huruf kecilnya harus dipenuhi semua. Untuk post test nanti Prof. Eka akan ada special surprise untuk mahasiswa-mahasiswa yang berhasil menjawab pertanyaan dengan cepat dan tepat. Baik, saya rasa ya Prof. Eka.
sebelumnya kita foto bersama sekali, boleh nggak foto bersama?
Ya Prof. Baik Prof.
Foto bersama ini bukan kita nanti. Tadi foto ini saya akan kirim ke orang-orang penting dengan pesan stop rapid test dan ini aksinya loh ya, semua setuju ya. Jadi ayo kita foto bersama. Siapa yang foto? Dr. Stephen atau siapa?
Baik, Prof, izin saya yang foto, boleh tolong dinyalakan videonya masing-masing. Jadi mungkin fotonya akan beberapa kali ya. Untuk yang pertama, 1, 2, 3. Sekali lagi, Prof. Eka, Prof. Astar, Prof. Iris, 1, 2, 3. Kemudian untuk yang halaman berikutnya, 1, 2, 3. Sekali lagi, 1, 2, 3. Sekali lagi, 1, 2, 3. Ini ada banyak, Prof. Ada 14 halaman, Prof. 1, 2, 3. Berarti mungkin, oke, yang ada fotonya, yang di halaman-halaman depan sudah saya foto, Prof. Yang ada videonya, Prof.
Jadi setuju ya, nanti yang ada di foto ini berarti ikut tanggung jawab bahwa kita setuju. Ya.
Setuju. Setuju Prof. Eka.
Tapi provokatornya cuma Prof. Eka ya. Provokatornya cuma satu ini.
Nanti kalau ditangkap jangan aku doang loh ya.
Saya yang mendorong Anda dari belakang.
Terima kasih.
Oke.
Steven.
Ditangkap duluan, gitu maksudnya.
Udah
Ya Prof, baik Prof, dari saya sekian sih Prof. Jangan lupa untuk ininya untuk mengisi form untuk e-certificate. Sebentar saya copy buat peserta yang ada di Zoom ya supaya bisa isi. Kalau untuk peserta yang ada di YouTube, untuk linknya nanti kita akan kirimkan ke email masing-masing ya. Sebentar ya, Prof. Mohon maaf tadi ada sedikit kesalahan teknis. Baik, sudah saya post untuk linknya yang ada di grup Zoom. Baik, saya rasa sekian dari saya, Prof. Eka, Prof. Iris, dan Prof. Astar. Terima kasih atas kehadirannya. Izin Prof, kira-kira apakah boleh kita tutup acaranya, Prof?
Oke.
Baik, ya, baik. Terima kasih, Prof. Prof. Iris, Prof. Eka, Prof. Astar, dan juga para undangan sekalian. Terima kasih atas kehadirannya. Sekian acara kuliah tamu kami. Sekali lagi terima kasih. Selamat siang.
Maaf, ngisi questionnaire yang ada di link tadi itu ya, yang Dr. Stephen kasih di link.
Ya, Dok.
Ya, terima kasih.
Ya, silakan,
Stephen,
Dok.
maaf.
Ya.
Nggak aktif linknya.
Linknya nggak
sebentar
bisa
dok.
dibuka, Dok.
Setelah acara ini ditutup baru saya aktifkan dok. Sebentar ya dok.
Oh, ya baik.
Ini memang belum diaktifkan karena acaranya belum ditutup tadi dok. Sebentar saya aktifkan semua ya dok, linknya dok.
Terima kasih Dr. Stephen.
Ya, terima kasih dokter. Terima kasih atas kehadirannya.
Dr. Stephen, makasih.
Ya, terima kasih
Dr.
dok.
Stephen.
Ya dok.
Terima kasih,
Dr.
Stephen. Salam sehat.
Terima kasih.
Ya dok, terima kasih. Terima kasih sekali sudah mau hadir.
Terima kasih Dr. Stephen.
Terima kasih dok.
Terima kasih dok sudah mau hadir dok.
Dr. Stephen.
Prof Eka
Ya, Prof.
dan moderator, luar biasa acara ini. Dari Makassar banyak yang hadir.
Nggak lupa.
Ya. Assalamualaikum, sudah oke. Leave saja deh.
Untuk linknya sudah saya aktifkan dokter-dokter sekalian. Kalau yang mau mengisi boleh ya. Terima kasih.
Terima kasih.
Ini bisa ditutup dulu baru kita ke link ya.
Kayaknya mesti di copy dok, karena kalau ditutup berarti keluar dok.
Udah.
Diklik langsung juga bisa dok.
Iya, mesti diklik langsung dok.
Oke. Oh gitu. Oke.
Dr. Stephen, kalau emailnya Prof Astar bisa dapat.
Emailnya Prof Astar saya sendiri belum punya dok. Jadi biasanya kontaknya beliau via WA, tapi nanti mungkin akan coba kita tanyakan dok apakah beliau bersedia atau tidak untuk emailnya kita bagikan kepada yang meminta dok. Tapi biasanya dok karena beliau banyak author dok, biasanya sih email institusi itu mudah dicari dok.
Oh iya.
Iya.
Oke, baik. Terima kasih.
Ya, baik. Untuk linknya sudah saya aktifkan. Jadi kalau misalnya yang mau isi untuk e-certificate bisa langsung mengklik linknya, atau juga untuk yang mau mengikuti post-test juga bisa langsung diklik linknya. Baik.
Baik, terima kasih.
Post-test kita harus ikut apa buat mahasiswa? Atau buat
buat mahasiswa, atau kalau dokter mau mencoba boleh dok. Untuk melihat comprehension-nya dok.
Sip.
Terima kasih Prof Eka. Dr. Stephen, linknya sudah ya?
Sudah dok, ini saya post lagi ya dok.
Sudah,
bisa dilihat di chat dok.
Ya, udah saya klik.
Ya mungkin ketimpa tadi soalnya udah di atas dok.
Kalau yang dosen bagaimana nggih, Dr. Stephen?
Kalau yang dosen silakan isi untuk yang e-certificate aja dok. Nanti akan kami proses dok. Tapi kalau dokter mau isi post-test juga boleh dok. Tapi tidak wajib dok. Yang wajib mahasiswa kedokteran dok.
Maksud saya kan ini ada pengisian NIM.
Oh, kalau yang dosen masukin ini aja dok. Nomor induk dosen dok.
Oh. Terima kasih Dr. Stephen.
Terima kasih dok. Terima kasih atas kehadirannya.
Sama-sama.
Because it has a very high sensitivity for diagnosis and it's a free of radiation that we can.
Terima kasih Dr. Stephen.
Ya, terima kasih dok. Terima kasih dok.
Dokter, bisa nggak ini diadakan rutin?
Rutin gimana nih Mas?
Minimal sebulan sekali. Kita ngundang teman seperti ini gitu.
Ya dok, nanti
misalnya setiap satu pagi deh. Kalau ini kan lumayan untuk pencerahan kita semua. Nggak usah mahasiswa, dokter-dokter juga lumayan untuk pencerahan.
Baik,
sebenarnya topiknya nggak usah jauh-jauh. Kita spesifik di COVID saja itu sudah cukup banyak. Misalnya tadi ini sudah dipancing beberapa. Contohnya itu tadi ada pertanyaan tentang DHF. Jadi ada yang rapidnya yang DHF rapidnya juga ikut positif juga gitu contohnya. Kemudian darah mana yang lebih bagus, darah vena, darah ini, bagaimana hasil studinya itu bisa jadi kita spesifik untuk COVID saja. Setiap Sabtu pagi gitu. Jadi untuk pencerahan kita semua. Jadi
Baik,
nanti
Dok, kita...
misinya Prof Eka untuk jangan pakai rapid itu bergaung, bukan cuman hari ini aja gitu.
Baik, Dok, terima kasih atas sarannya, Dok.
Ya. Makasih Dr. Stephen.
Makasih,
Semoga
Dok.
bisa Sabtu depan ketemu lagi ya.
Makasih, Dok.
Yo.
Ya, saya Dr. Stephen dari Makassar.
Ya, Dok.
Ya, ini saya tadi setuju banget dengan usul itu, tapi mungkin kita kasih spesifik ya, misalnya Dok ya, innate immunity, misalnya innate immunity dulu dibahas, kemudian habis itu humoral immunity, seperti itu. Jadi ada pembelajaran berkelanjutan mengenai imunologi, ya.
Baik, Dok, nanti kita coba sampaikan, Dok.
COVID ya, mengenai COVID.
Ya, terima kasih, Dok.
Ya.
Baik, Dok.
Ya, saya Peralumni Makassar, Dok, ya, dari Unhas.
Ya, Dok.
Terima kasih.
Terima kasih, Dok.
Sometimes we found that seem not severe, not cyanotic vein, but actually it's diffuse cyanotic over the ID vein. And when you check with IBS, you see some patients suffer from this working pesticide, which might give you wrong information. And for the size eating, we
tanggal berapa? Oh, minggu depan ya.